participants joining at that point. Okay, so just to make you aware that this session is being recorded, hopefully you've all seen that box come up and you have accepted that. So just so you all know that we are recording this session. So this session is part of the NIHR statistics group. And so what we're really trying to do is bridge that gap between statistical methods and application into research. So we're trying to not just appeal to statisticians, but appeal to people that use statistical application in their work. So there are several working groups as part of the NIHR statistics group. We are the laboratory studies group, and we have other courses and sessions that we are running. So I'll give you some information about how you can keep up to date with that if you wish to. So if you wish to participate in any future courses, or at least be aware of them, and you can see if you would like to be involved. So today's session then, hopefully you've all seen this as part of the advertising of the session, but we have 3 really good speakers today, and we also have a chance for some networking. So we have a breakout room session at the end of the day where that should be an opportunity for people to ask more questions and talk to each other. I know it's a little bit difficult when we do these things on Zoom, so we've tried to build some of that in for you as well. If you have questions, please use the chat function, and there will be some time for questions as we go through the sessions as well. So if you use the chat function to just make sure they're recorded there, then we can use those when we go back to that. And as we've said, it'll be recorded, and we will try to make this available via our NIHR statistics group page as soon as possible. Probably be linked out through to YouTube, I believe, but we'll make you aware when that is up and running. So we do have a mailing list that you can join. So this is a mailing list that you can sign up to, and then you can decide if you want to leave it. So you have complete control over that. I will put that into the chat function, so you've got the link there if anybody wants to take the opportunity to join that today. And it's also available from the NIHR Stats Group website as well. Okay, so that's all I wanted to do in terms of introducing you to the session. So I will now hand over to Jeremy, who's going to lead us in this session today.
Thank you, Alice, and thank you everybody for joining this webinar. So I am one of today's speakers. So I will introduce myself just before my talk, so make sure you don't miss that one. Let me now introduce to you our first speaker, Mallory Freeberg. Mallory holds a PhD in bioinformatics from the University of Michigan, and now works at the EBI in Cambridge as the coordinator for the European Genome Phenome Archive, in short, EGA. She has a long experience of supporting high throughput molecular data, curation, submission, archiving, and distribution. During her time at EBI, she coordinated submissions of the first single-cell RNA-seq data from the Human Cell Atlas, and led archiving and distribution of the whole genome sequencing data from UK Biobank. Mallory, it is a real pleasure to welcome you in this workshop. Without any further ado, over to you.
Thank you so much, Jeremy, and thank you to the organizers for inviting me to present at the workshop today. I'm going to share my screen, get all set up. Excellent. So can everyone see my title screen?
Yes, we can.
Excellent. So as mentioned, my name is Mallory Freeberg. I am the coordinator for the European Genome Phenome Archive, which is one of the resources at EBI. And when I was invited to present to this group at this workshop, I decided to take a bit of a broad approach to the larger topic of accessing and analyzing high throughput genomic data from public repositories and kind of frame it from the perspective of FAIR principles and generally promoting openness in the life sciences, but I'll also take us down to the specifics to different genomics topics throughout my talk. I'm fairly sure most everyone here has heard of the FAIR principles. So this is an acronym that stands for findability, accessibility, interoperability, and reusability, which are principles to help optimize the reuse value of generally all research outputs, but specifically these principles were designed to initially review data outputs. So findability essentially means that our data are available in a public repository and are assigned accessions that we can then reference to find or discover the data of interest. Being accessible means that researchers can download the data or otherwise use it in reanalyses, for example. Being interoperable means the data are represented in standards that can be used by different tools and different platforms. And there's sufficient metadata describing the data that use data vocabularies and ontologies, which are concepts that I'll talk about later as well. And finally, reusability is probably one of the most important. It means not only can we get to and download the data of interest, but we can actually understand it. So there's enough metadata around these research outputs for us to reuse it. The bottom left of my slide here are a few more links to some other resources related to the FAIR principles. I have lots of extra resources linked throughout my slides, which I will happily share after my talk. I'm not going to go too deep into any one of the things I'll talk about, but if something piques your interest or you have more questions, there are going to be lots of links to find other resources about the topics. So why should we care about being fair? Well, I already mentioned that this is really a way to maximize the value of not just data research outputs, but all of our research outputs. We put so much time and energy and effort and money into generating research data and all kinds of outputs that it really makes sense for us to be able to maximize the reuse value, not only with close collaborators, but really all of the scientific communities that we are a part of. And this feeds into being generally good stewards of our scientific communities. And then to further promote scientific progress, I think most of us either partially or maybe majorly are in the research or medical fields because we really just want to make our world a better place. And so this is why we should care about thinking about these FAIR principles. I also wanted to quickly mention that I will be talking about FAIR a lot, but fairness doesn't always necessarily equal value when we talk about research data. And this is just a point to keep in mind because although we strive to be fair, it's not a binary. It's definitely a spectrum. And you can be in different places on the spectrum in different parts of your research. And in fact, you can have data or research outputs that are not very fair, but still have immense value to your community. But just to recognize that this means there will be extra barriers to enabling people to reuse that, that data. And then even a very fair data resource or research outputs can sometimes be of low value to the scientific community if they're not reused in further research. So just something to keep in mind today. So thinking about the FAIR principles, the major way we think about these is as they apply to our data and metadata research outputs. But in fact, I'll touch upon a few more areas where we can apply the FAIR principles, such as research code and software, computational and experimental workflows, publications and other digital objects, as well as training materials, which I think is probably the topic here that people think of least in terms of having research or having value. But in fact, it can encompass all of the other things I've talked about and add a lot of extra value to the data that you're putting out there. So I'll have a bit to say on training materials as well later. So if we focus on, again, data and metadata in terms of FAIR principles, in the field of genomics and transcriptomics, I think the most obvious way we think about this is in terms of the file formats that we use to both share and reanalyze data outputs. So for sequencing reads, this is of course formats like FASTQ. are alignment formats, BAM, CRAM, and SAM. And recently, with the advent of newer technologies like nanopore sequencing and some of the more complex data structures, we are starting to see formats such as the FAST5, or you might also see this as HDF5 for having a more hierarchical representation of this type of data. Oftentimes, when we have these sequencing outputs, the first thing we want to do is understand the variation between our samples and some kind of reference. So to represent those variants, we have the variant call format VCF and its binary counterpart BCF, which are some of the more standard formats. And it's also worth noting to be thinking about not only sharing these data files, but also their associated index files. So when thinking about reusing these types of data, often we require indexes to do the analyses. And so it's a good idea to submit these along with your data files. It can also serve as a way to do sort of a quick QC on the data before you archive them. In the context of this group, I think many people are probably doing human-related research. So there's two interesting emerging format standards I wanted to mention that are more useful in the human or clinical research areas. And the first is for sharing disease and phenotypic information. This is called the PhenoPackets standard. This is a standard that is very structured, has lots of ways to not only share disease and phenotype information, but also pedigree information, demographic information, clinical results. It's only a few years old. I think the first version was confirmed in 2019. But it's already been shown to be really useful in contexts such as rare disease research. The second is an encryption format. So oftentimes when we're dealing with human data, we need to have the data files be encrypted in various stages to prevent unauthorized access. We've been encrypting data files for many years, and there's a new format, this crypt4 GH format, which is a more efficient way to do the file encryption, which still enables reanalysis. And you'll notice that some of these formats have highlighted in blue and bolded. They are formats that have been, that are maintained and developed by a global community called the Global Alliance for Genomics and Health, or GA4GH. And this is a group of researchers across the world who have really understood that it's important to have standards in terms of implementations and policy frameworks and other aspects around sharing human genetic and health data. And so these are, again, some of the formats that this group is maintaining. Of course, we also can think about metadata standards. So the first two I've listed here, the Miami and MinSeq, are probably the most well-known for microarray and sequencing experiments respectively. And these sort of define the minimum information that's required in order to understand these types of experiments and then potentially reproduce an experiment to get similar results. These formats these standards are quite old. I've also listed two formats or standards for metabolomics and proteomics, which you might also be aware of. And then the last two I've listed are getting into this area of having standards, not just for an experiment type, but for a particular domain of research. So the bottom left is a standard for functional annotation of animal genomes. And the bottom right is a standard that came out a couple of years ago for understanding single-cell RNA-seq experiments. And just a caveat, I helped create this single-cell RNA-seq standard, so I'm quite familiar with it and happy to chat about it later at the breakout. Okay, we're actually going to do a bit of an interactive session for the next couple of minutes. So if everyone can open their chat window, and I'm going to request you to put in some answers to this question. We'll kind of see how it pans out. So let's say that you're doing an experiment and you're collecting metadata for your experiment, and you're asked to provide a metadata value for the sex attribute to describe the fact that you're working with a female sample. So in the chat, just drop in what value you would put if you were asked to enter this metadata value. And you can put one answer, you can put multiple answers. We'll just give everyone a couple minutes to put what they think they would put here in the chat, and we'll see what we come up with. Or people might be very shy. Okay, I see binary 2, 0, 0, 1, F. This is great. Just as many different answers as there are participants. This is probably my most favorite slide that I have to admit I stole from a colleague who worked with the ENA maybe 5 or 6 years ago. The slide is old, and she did a curation of this attribute for samples submitted to the European Nucleotide Archive, the ENA. And this is what she got. This is what she found by just doing a simple look at what people were putting for this value. And you can see, first of all, lots of synonyms, some typos. There are some terms here that are specific for a particular organism. And you can even look at the last one, someone put female and then gave like a whole big note describing what actually happened with their sample. But the point of the slide is that there's clearly a need for us to have standardized ways to represent the same concepts. And this is where ontologies and controlled vocabularies come in, which can help alleviate having misunderstandings or lack of clarity of metadata terms. And thank you everyone for participating. That was really fun to see what people put. And so I'm thinking about what archives we already have available that are quite popular in the genomics and transcriptomics arena for sharing these types of data. So the 3 main public archives for nucleotide data are the SRA with NCBI, the ENA, which is at EBI, and then the DDBJ in Japan has a sequence read archive as well. And then the next row are sort of the corresponding archives, but for controlled access human data. So NCBI has dbGaP, the EBI has the EGA, and then there's a Japanese genotype and phenotype archive. And these are mainly for sort of like primary sequence data, but these archives also take some forms of process data and have their own metadata standards. And then along the bottom of the slide are a couple of the key archives for sharing functional genomics, expression and variation data. You've probably heard of at least some of these. So there's Array Express, Single Cell Expression Atlas, and the EVA are all at the EBI. And then there's Geo and dbSNP with NCBI, and then Japan has the Gene Expression Archive. And so depending on what kind of data you're looking for, any of these archives are probably going to have some data that may be relevant for your research. I was thinking about the best way to describe how to actually access data amongst all those archives. But I definitely won't have time to go through using all the different interfaces, but I wanted to just give a breakdown of what the key steps are. So first is the step we actually often forget about, and that's the discovery step. And so most of these archives will have web-based browsers that might allow basic or more advanced querying, that you can look for data related to maybe the disease that you're studying or an organism you're studying. Sometimes they might have more programmatic search functions. But after finding data of interest, it then becomes an issue of accessing the data. So for most of the public archives, data can either be accessed anonymously, or you might have to just create an account so they can track who is accessing the data. And then for the human controlled access archives, you will typically need to create an account and then apply for access and be approved by the appropriate data controller. The next step is actually downloading the data. This can be done a number of ways. Some of the archives have bespoke tools to access the data. Sometimes they use standard protocols, standard transfer protocols, such as FTP or Aspera. And some archives will have maybe web-based browsers for enabling download of smaller data sets. And then finally, the point where probably there's most divergence is in doing analysis of the data that you now have access to and have downloaded. Oftentimes, the simple way is to download the data locally, and you can run your workflows on your computer, or maybe you have a cluster. If you're working at a big research institution or organization, there might be an institutional cluster that you can submit jobs to and run your analyses. But really where the field of genomics is kind of going, is already going in this direction and will continue to go, is using these bigger cloud-based platforms, or you might hear this term research environments, which can be, again, shared amongst an institution It's really becoming more prohibitive these days to move the data from place to place because it's so large. So it's easier to have a single copy somewhere, usually in a cloud, so that you can bring your analyses to the data and run them there. And because I work with EBI, I just also wanted to shout out to a lot of the other resources that we have outside of just the archives for genomics and transcriptomics. A lot of archives and resources for chemical biology, for protein research, a lot of other molecular systems, so interaction databases, all kinds of portals for exploring data in different ways. So if any of those other fields are things that you're interested in, there's lots of resources at EBI that you could take a look at. So getting into some best practices for ensuring our data are fair, there's really a recent paper that sums this up quite nicely. It's one of those 10 simple rules papers. It's for annotating sequencing experiments, but really this can be applied to any type of research that you're doing. And the journey kind of starts probably earlier than you would think, which is before you even start collecting samples or doing your experiment. It's really important to be thinking about what you ultimately are going to be sharing with people and the best way to collect the appropriate metadata and ensure that your data are fair when it be shared. It's also important at this stage to define the roles. So having someone whose role it is to ensure that everything is being collected and standardized correctly is quite useful. And that way, when you start to actually generate data, you can already have an idea of what standards you want to follow, whether it's a experiment type standard or a field or domain specific standard, understanding what metadata that you want to collect, what ontologies and controlled vocabularies you would like to use, which are relevant for example, if you're studying rare diseases. And then also, especially in human studies, it's important to understand the legal restrictions there might be in place for what information can be shared and whether it has to be anonymized or pseudo-anonymized. These are important things to keep in mind as well. And then finally, this is probably the point where most people start to think about these concepts, is getting ready to either share data with colleagues or publish in an archive in preparation for publishing an academic paper. And at this point, you've done most of the work. You can do quality checks before actually submitting your data to an archive. Having data freezes is a good idea if you're sharing analysis results, because we know analysis results can change with the changing of any parameters of your workflows. So this general timeline, again, although it was specific for sequencing experiments, I think it can really be applied to any sort of research concept. Also, since the publishing of the FAIR principles, there's been a lot of work, a lot of initiatives to help people understand both how fair they are and what they could do to become more fair. This is just a smattering of the ones that I've either been directly involved in or I know of, and I've put different links. There's websites and tools that you can look at. But really, if this is an area where you kind of want to dig a bit more, I highly recommend looking at some of these self-assessment tools, especially early on in your research planning. The FAIR Cookbook is an especially interesting resource that has lots of tips for figuring out how to make your research outputs more fair. But any of them would be useful. Okay, I mentioned at the top of my talk that I'm also going to review how other research outputs besides data can be fair. And so I'll be talking next about the publications, code and software, training materials, and workflows. And I'm kind of just giving like a tasting of all of these. And if you are curious about any one of them, there's lots of links here to different resources. So code and software, research code and software is probably the next biggest area that there are efforts to ensure that these are fair. I think it was last year, the same sort of working group that initially developed the FAIR principles for data came out with a FAIR principles for research software paper. So there's a link there you can go and read. But there are some very simple things that we can do to ensure fair code and software, such as making sure that we're including the correct licensing for reuse and adaption, having README files so we can understand what the code is actually doing, ensuring we're using a versioning control system to manage the code. And then it also becomes difficult if we are using someone else's code or software and there is not an associated publication. We often don't cite that software because there's not really a clear way to do it. So there's been an initiative to develop this citation file format, which is a standard way to cite any code or software that has not been published. And so if you're in the field of producing code and software as one of your main research outputs, this is a good standard to look into. For workflows, there's a nice paper on making computational workflows fair that I recommend looking into, or using something like a Jupyter notebook to record what computational pipelines you've run and putting in explanations of how to use parameters. This is a nice tool to have, and I find that when research outputs are released with the Jupyter notebook, it's really easy to sort of like dive right in and start working with the workflows. And finally, this protocols.io is a really, really cool initiative that started, again, not that long ago, maybe 5 or 6 years. And it's a way to share not only computational workflows, but also actually started out as an experimental workflow sort of sharing platform. It's almost like a merger between a lab notebook, electronic lab notebook, and GitHub. So people can share any kind of workflows. Sometimes they come with checklists, so you can actually be doing the experimental workflow with this open and kind of check off as you do the steps. People can comment, ask questions. There's version control. It has different communities. So I know there's like a single cell experiment community within protocols.io, so you can find all of experiments that have been published there. And it also is a pretty fun website. I think there's like a protocol for how to make a sandwich. Sometimes they have some really like interesting topics that's just kind of like fun to look around. Next is publications and other digital objects. And I don't mean academic journal publications, but I mean anything else you produce that doesn't really fit into maybe a category, but you want to make sure has a citation record and you can reference later. So Zenodo and Faculty 1000 Research are great resources for this. You can find lots of things like slide decks, webinars, reports. I think you can also publish training materials and small data sets in Zenodo as well, which is quite useful. And of course, I will advocate and shout out for preprint servers as a good way to both share and find sort of pre-publication research outputs that might be useful. And then finally, I'll mention the training materials. There's a nice 10 simple rules paper and an associated webinar if you want to start thinking about developing training materials to be published alongside of your research and how we can make that fair. And then also an interesting resource, if you're familiar with the Galaxy project, which is a web-based platform for enabling sort of non-computational folks to do very complex bioinformatics pipelines and workflows. That project has its own training network. So if you go to their website, they have all kinds of training materials for transcriptomics, genomics, proteomics. They have climate science tutorials, and they even have, I think, a set of SARS-CoV-2, COVID-19 tutorials that came out recently. So that's a nice place to start looking at how other people are doing different genomics and transcriptomics workflows. Okay, so this is my last section, and I'm just going to do kind of like a bit of a dive into the EGA and how we exemplify some of the principles that I've discussed so far. So the European Genome Phenome Archive is a permanent archive for sharing all types of identifiable genetic, phenotypic, and also starting some clinical data. This has largely been driven by data outputs from biomedical research projects, but we're seeing an uptick in this type of data being generated through national healthcare initiatives, which is another interesting topic that I'm happy to talk more about. Everything archived at the EGA is assigned a permanent and unique accession. So on the left here is a table of the sort of headers we assign to different types of objects, and they all get a unique number as well. And these can be referenced in publications, for example. So someone who's reading your paper and wants to perhaps reanalyze your data, they can find the accession and come to the EGA website and find the data that they're interested in. We also have a metadata standard, which is quite interoperable with the ENA standard. We represent ours in a particular format called XML, which is what's shown on the right. You don't actually have to understand any of it, but just to know that this is how we represent what are the minimal attributes that we require for submissions to the EGA. It also defines some of the optional attributes and encodes some of our validation rules. We use a lot of control vocabularies and ontologies. On the right here is the list of Illumina sequencing machines. This is a controlled vocabulary list that we update as new machine models become available. And on the samples, we collect phenotype metadata. And from this, we request that submitters take terms from the experimental factor ontology, which I just showed a screenshot of what that looks like on the left here. We also use an interesting and new ontology that actually came out of the Global Alliance, the GA4GH I mentioned previously. This is an ontology of data use conditions and restrictions, and this is especially important in human research fields, and it defines any conditions or restrictions on how the data can or cannot be reused. This is a screenshot of a data set that's at the EGA for which it has specific data use conditions. And the table at the bottom here, you can see, for example, this data set has a general research use, which typically means it can't be then used for commercial purposes. And it has some restrictions on the institution and the user, the particular person who can reuse the data, and it does require follow-up publication if there are any results from reusing this data set. We've also started implementing the PhenoPacket standard that I mentioned earlier for representing clinical and pedigree and demographic information. We've been working closely with some rare disease research groups to sort of pilot this PhenoPacket format, and it's quite interoperable with other resources and tools that are also interested in using this particular standard. So just another way that we're both, driving the development of and adopting the use of some of these more complex human data related standards. Okay, so with my last slide, I just wanted to mention a few other topics related to what I've been talking about, but didn't quite have a place to put them in my presentation that I think if you're more interested, I highly recommend looking at. The first is a mentoring and training program called Open Life Sciences, which is great if you want to maybe dig a bit deeper into how you can bring in some of these more open practices into your research group. And there's a website here all about how this training program works. In parallel with the FAIR principles, it was either last year or the year before, this other set of principles came out, these CARE principles, specifically for optimizing the reuse value of indigenous data, which is also something that's really important to consider if you're particularly working with these groups of people. So there's a paper here and a website where you can learn more about the CARE principles. And then I put a few more links here for resources for making fair research code and software, and also some more on ontologies as well. So with that, I'm happy to have time for questions, and thank you so much for your attention.
Thank you. Thank you, Mallory. People can raise a hand for question. I have one. Can you tell us more about the controlled access? So you put in contact the data owners and those wishing to access them, and they also sign a contract. Is it how it works?
Yes. So in the EGA's specific example, we don't own the data. It's always the person who has submitted the data to us who, in GDPR terms, they are the data controller. And so we sort of like mediate that interaction. So if you are interested in downloading data from the EGA, you actually get in contact with the data controller, and we put contact information on our website, and it's between that data, the data controller and the requester. Usually there's some kind of data access agreement. There might be other terms for it, data transfer agreement, but there's usually some agreement that must be signed with the different terms of use. And only after that agreement has been reached, then the requester can come back to us and say, I was approved, can I access the data now? So there is an agreement that has to be signed before the data can be accessed.
Yeah, okay. Any other question? There's one in the chat asking if there is a recommendation or a guideline for coding the variable sex or gender.
Yes, I love this question. I think in general, we have been very sloppy in how we, meaning the scientific community, have been very sloppy in how we've represented this in the past. I've seen a couple different ways to be more specific about this. So oftentimes when we see like sex or gender, I feel like in especially the nucleotide archives, we're talking about like a chromosomal sex. Like what are the chromosomal indications of that sample? Because when we're talking about genomics, like that's kind of what we think about. So I've seen sometimes the attribute say biological sex, and then the interpretation is chromosomal, like chromosomally, what was this individual? But even that is not entirely accurate, because there's lots of other ways that we tend to classify something but in a binary way that's not actually binary. So I don't think there is an official classification, but the difference between biological sex and then gender, I think, is one way that we've started to tease apart this. But also being very explicit, like if you actually want the chromosomal representation of an individual, then you just call the attribute, chromosomal sex or karyotype or something to mean that. But I don't think there's actually an official or a totally agreed upon way, but I think we're getting better at teasing apart what we mean and also understanding that it really is all a spectrum anyway. So it's not maybe a satisfying answer, but I think we're getting better at understanding how to represent those values.
Yeah, the difficulty and the lack of common standards for metadata is really an issue, and will be covered more in my talk, which is the last one. So make sure you don't leave too early. Any other question? So Mallory, the EBI has both these controlled and free access data type. What do you reckon the users think about? Any feedback from them? Which one do they tend to prefer?
It's always a much longer process to get access to controlled access data. But the only controlled access data at the EBI at the moment is with the EGA, because we're the only resource that has potentially identifiable human data. All of the other resources are at a point where, for example, proteomics, we don't quite think a proteomics data set about an individual is identifiable yet. I think this is changing. I think we're seeing that because we're integrating so many omics data sets that it might actually become possible to re-identify someone with something other than a genomic sequence. So although all the other archives are now public and it's much easier to get access to public data, I think we'll start to see some of the resources start to actually have some controlled access data sets because they have the potential to be identifiable. But really it's a time and effort barrier for getting access to the controlled data, but we have to respect the consents of the original individuals who contributed their sample to generate the data. So there's not really a way around that at this point.
Okay, great. Kevin has one.
Yeah, that's an interesting question actually, Mallory, about is RNA sequencing data considered identifiable? Because often you could identify SNPs and things from that type of data, or is that typically viewed differently?
At the moment, well, at least at EGA, it's still considered identifiable because, as you say, if you get enough information and if there are enough rare SNPs perhaps, or if you have a combination of certain SNPs from the RNA-seq and some other demographic information, then you start to be getting closer to passing the threshold of being re-identifiable. But that whole threshold question is a whole other research topic that we could discuss about how much information combined gets you past that re-identification threshold.
Okay, many thanks. And we are now moving to our second speaker. I have known Kevin Litchfield for many years, from the time we were both members of the Testicular Cancer Consortium called simply TCAC. Kevin holds a PhD in bioinformatics from the Institute for Cancer Research in London. He is currently a team leader at the University College London Cancer Institute, where he conducts research focusing on cancer immunogenomics and immunosurveillance. Kevin has a track record of analyzing large-scale molecular data, both those generated in-house and those downloaded from public repositories. So thank you very much, Kevin, for accepting to talk us through some of your experiences of dealing with these large data sets. Over to you.
Thank you, Jeremy. Yeah, let me share my screen. Okay, so hopefully that looks good. So yeah, no, thanks to the committee and Jeremy. I was delighted actually to see this topic, because it is exploiting and utilizing publicly available sequencing data is something we've found tremendous value in our recent academic work and also going forward. So it's a topic close to our heart, and I think everyone really should try and exploit and get the most they can out of this vast array of data that's now available. So just to go through the agenda for my talk, so we start with some brief background and introduction to some of the concepts, then to go into some case studies that we found great benefit from accessing and using publicly available data, and tips and ideas perhaps for others that might find useful, and then go in depth on a particular case study of a recent meta-analysis we did, again exploiting publicly available data, and then a bit of time at the end for questions. So it's stating the obvious, but just to show it, in graphical form, the growth of biological data here, for example, DNA sequencing data has probably outstripped anybody's expectations here. You can see the growth rate of Illumina DNA sequencing. It's obviously grown quicker than Moore's law, or even quicker than Illumina's own expectations of how usage growth. And this probably is the same across many technologies, even things like single-cell RNA sequencing with 10x. Again, it's probably grown quicker than anyone could have imagined, even the companies that have designed and make profit from these platforms. Here's a quick snapshot in GEO, Gene Expression Omnibus. Here you can just, millions, 4.5 million samples, they're available, over 2 million of them human. So in pretty much any question, or many genes, many questions, many examples, there may be data that's been generated that could immediately address a question and save a wet lab team time and effort from redoing something that's been already done. Single cell data, as I mentioned, people use that now to understand more deeply biological patterns in individual cells here. Again, this has only really been initiated fairly recently, but we've already got nearly 14 million cells in the human Cell Atlas portal from thousands of different specimens and donors, all immediately available. One philosophical point, I guess, in terms of this usage of data that's been already available. Many people might remember the New England Journal of Medicine editorial about data parasites that was heavily criticized, and I guess the authors of that, probably may have regretted those comments. Those comments were potentially badly phrased. But I think certainly every interaction I have, I never forget the fact that as I haven't generated the data, I haven't done all of the work, and really there's huge sort of appreciation and gratitude back to the collaborators that do generate the data. I think data should be reused. Investigators don't own the data. If there's data generated in my lab, I don't own that. Really, the data is only owned by the patient, if anybody. So it's free to be reused, but certainly the effort that goes into generating it, I always try and bear in mind and be as collaborative as possible, and where appropriate, acknowledge through authorship if people have generated data. So really, I guess the best way I think about it is in terms of upcycling of data. So, a data may have served one purpose, but that doesn't mean that it's the end of the road for that data set. It can be upcycled, reused to generate new knowledge. And as data sets grow and become more complex, but also more interlinked and high dimensional, the opportunities for upcycling really become more and more exciting. So just to summarize the background there, I think as anyone would know, that the availability of public data now is really unbelievable and growing very rapidly. It's not just genomics data, as I'll come on to in some of the examples, as rich proteomics data, imaging data, flow cytometry data, whatever platform you work with or whatever research questions you've got, there's putting more data out there than you would expect. Obviously, this is only going to increase further as most funders demand that data is made publicly available, at least charitable funders for not-for-profit reuse. And so I think to be a competitive researcher, really, you need to know what data is out there, how to get it, and how it can be used to further your research. But not forgetting that, respect is critical to the original data generators. So just to go into some case studies. So one particular recent case study we had in our lab, we're very interested in a particular pathway, the nonsense mediated decay pathway, that's involved in mRNA quality control. One of the key genes in that pathway here, UPF1. We were about to undertake some knockdown experiments, but we also need to design some primers, and we wanted to really try and get a head start and get some testing and optimization done before we managed to do our own knockdown, generate our own RNA-seq data. We're able to immediately go here to the gene expression omnibus, just put in simple keyword search, already find immediately the second result there returned from that search was an interesting looking data set. We look further at this data set, exactly what we needed, RNA-seq, generated by actually a very reputable lab in the nonsense mediated decay field up in Edinburgh. We look further here into the data. We saw it's ideal, exactly the gene we wanted to knock down, and not just triplicate, 5 different replicates done for this experiment. We're able to quickly go and get that, download the FASTQ files from that data set, and then run our pipeline. We knew what we wanted to do with the data. Our pipeline was ready. We wanted to look for particular isoforms that were differentially expressed upon knockdown when that pathway was inhibited, to see new isoforms that were no longer degraded. And so we were able to do our standard type of analysis, find some really good markers here. Sorry, the quality is not very good, but you can see upon knockdown here in the lighter color, we had a whole range of markers that were consistently elevated and at very low background levels in the control samples. And this all really took a day or so or less in terms of the analytical time. And then we could then straight away take in our own cell lines, our own experiments, convert those markers into simple qPCR assays, and within a few weeks really knock down and understand the importance of our target in a range of different cancer cell lines using this panel of 7 or so different markers. In another situation here, in terms of proteomics, you're interested in resistance to alatinib, an EGFR inhibitor, which is very important in non-small cell lung cancer. Again, here in terms of proteomics, the PRIDE archive is a great repository of data. Again, a simple search, we're able to find here exactly the sort of data set we were looking at, a pre and then a resistance to treatment as imatinib, same sort of EGFR inhibitor. Post-resistance samples, we're able to immediately download that data and look for proteomic changes or causes of EGFR TKI resistance. Again, here another example, staying on lung cancer, one of our key tumor types of interest. Here we're interested in single cell data, here in terms of 10x, a major generator or platform 10x data. In their repository, you could again go and search for lung cancer, immediately found a data set of disseminated tumor cells from lung cancer patients, 7 different donors. So we're able to obtain and use that data. This time it's to generate preliminary data for a grant. It was, a quick, sort of easy way to make use of publicly available data. Also in gene expression omnibus, there's increasingly more and more single-cell RNA-seq data sets deposited. Again, just with pretty simple search terms, you can find relevant data sets here, one with both single-cell RNA-seq as well as site-seq in some patient non-small cell lung cancer samples. So as I said, it's not just really sequencing data. Like most labs, are increasingly trying to make use of imaging data, particularly technologies such as imaging mass cytometry. For those not familiar, Cytobank is a great platform for accessing available IMC data from top labs here, such as the Bowden Miller Lab, which have really led a lot of the IMC work, at least in cancer context. You can go and access data from their experiments and other labs. This is rich data. As you can see here, you can do single cell segmentation, look spatially at different cell types, their interactions with neighboring cells, how those groups of cells form into unique neighborhood patterns. So this data is, again, just really a few clicks away, very easy to access, to start to use. Also, I think a recent step change in the last few years has been the availability of NF-core, NextFlow pipelines. So even if you've never used in your group IMC data, it's pretty easy to find an NF-core pipeline. Here, this time, IMCyto, a pipeline that's exactly designed to analyze this type of data. You can get it downloaded and run through and start to get some results very quickly to evaluate whether this technology is right for your lab, or potentially get preliminary data to support, getting funding to take this sort of thing further. So going now to a more in-depth example of how our lab has recently utilized publicly available data to do an in-depth meta-analysis. So just to start with some background. So apologies for those statisticians on the call. This will be obviously very basic, but for those that are not statisticians, just to give some basic definition in terms of meta-analysis as a quantitative approach for systematically combining results of previous research to get new conclusions from the totality, the body of research. So historically, meta-analysis is really, I guess, attributed to initially Carl Pearson in his work, one of the first pioneers to use formal techniques for combining data in his work on looking at the efficacy of typhoid vaccination. And his rationale, which is as important today, I guess, as it was back then, is that often many groups, many studies are too small on their own to find a reliable opinion. But when they're combined together, you can start to minimize that error and arrive at more reliable conclusions. And this is exactly the reason really we undertook meta-analysis in our context for understanding immune checkpoint inhibitor response in the context of cancer immunotherapy treatment. So there's two different types of meta-analysis: aggregate data meta-analysis, where you take the summary point estimates from studies. So this can often be derived from supplementary tables or through dialogue with authors, but it's where data is not available for individual patients. Instead, you might take dozens or however many studies that have identified the effect sizes and take the data at that level, not going down to the individual patients. The other type of meta-analysis is where you can actually obtain individual patient level analysis for all those enrolled in each of the trials included in your study. And these have particular benefits. You can obviously standardize methods across each trial, but then also look for investigation of patient-led level modifying variables. And there's two different types of model that you can use in the meta-analysis. So a fixed effects model is where you assume that the effect size is the same in each study, and then any differences is solely due to random error. Or the other type of model is where you potentially expect there may be differences in effect size across the studies, maybe different cancer types or different disease stages. The effect size of a treatment or other variable of interest may actually be different. So you can allow for that inter-study variability. Here are the standard steps in a meta-analysis here. Start really by defining the research question and the hypotheses. It's important then to define the criteria for including and excluding studies. You then need to locate your research studies and then apply those criteria you defined above to determine which studies are eligible for inclusion. You then need to classify and encode the important study characteristics and length of follow-up, definition of outcome variables. You then need to select translate the results from each study using a common metric or the common metrics you can measure in each individual study. And then you begin the aggregation process of findings across studies to make your weighted pool estimates of effect size and then evaluate statistical homogeneity across the pooled studies. So just one example of a typical design from the literature here in arterial fibrillation study here, where authors had identified up to just over 1,100 potential articles. They then applied their search criteria here to deduplicate studies or remove studies based on different methods, based on different technologies, remove studies that weren't actually primary data, remove ones with different outcomes, remove ones with too small patients per group, as well as remove any retrospective analyses. That then dramatically reduces, and this will be typical, dramatically reduces the number of references. You can then do further exclusions here. The authors exclude based on inadequate reporting of efficacy and safety outcomes. They then have the number of studies that may be relevant. There's then some further duplication where there's multiple publications from the same data sets. They find unique data sets, they contact authors, they get some attrition there in terms of authors that either refuse participation or fail to respond. You then get the final data sets where available for individual patient level data. So moving to our example here, we're interested, as I mentioned, in biomarkers of immune checkpoint inhibitor response. Is a pretty recent modality in terms of cancer treatment. Over the last decade or so, it's become a major treatment type used in a dozen or so different cancer types, but most of the biomarker work in this area had been fairly underpowered, cohorts of 20, 30, 40, 50 patients. We wanted to try and attain a higher sample size, 1,000 or more patients, to try to eliminate some of the false positives that had accrued in the literature, as well as understand a bit more robustly what the drivers were of response to this type of treatment. And here the code for this work is all available on our GitHub there for those that may be interested. So we started with our set of available studies. Here is our study design. We found a dozen different studies where we were able to obtain raw sequencing data, individual patient level data. This was important in this case because we wanted to derive the biomarker values for every patient in a harmonized way from processing the raw FASTQ files. So we're able to obtain the sequencing data as well as harmonize clinical variables, process all that into a centralized database to then conduct the different types of analysis we were interested in. So here, touching on Mallory's examples here, we used extensively data from EGA here, several EGA data sets, as well as dbGaP data sets, the US equivalent for controlled access data. Some data sets were in just GEO themselves in an uncontrolled setting. And then some of the data sets we had to just directly obtain from the authors via email request. We here wanted to look broadly at biomarkers, so we wanted to have a systematic approach in terms of the biomarkers we were going to assess. So we defined a set of search terms here, all these different search terms for potential biomarkers for immunotherapy, the different types of immunotherapy we have, anti-PD-1 treatment, CTLA-4 treatment. So we had various combinations that we searched in PubMed with a particular date range. We did the search in 2020, so we had a criteria here. Within the last 5 years, articles were selected. This gave up to over 700 unique manuscripts that we further reviewed and subsetted for being based on human data. We didn't want to assess biomarkers only from preclinical data. We then excluded things like case reports, along with review articles, as well as biomarkers that we couldn't calculate in our exome or RNA sequencing data. And that gave us in total 55 biomarkers that were included in our meta-analysis. So here's just an overview of the key biomarkers we're able to put in the main figures in our manuscript. Here you can see we sort of laid out all the different biomarkers here as rows, and then all the different studies here as columns, able to quantify the effect size. We didn't want to try and quantify based on p-value. We focused on effect size, which we harmonized as radiological response. Here you can see here blue is patients or biomarkers that enriched for higher odds ratio of radiological response to checkpoint inhibitor treatment, and red is markers that enriched for lack of response. So you can see here with significant ones here, and we're able to quickly identify biomarkers that were important, and also biomarkers that didn't validate, and I'll come back to that a bit more in a second. But just first of all, to show some of the QC steps that were critical for this work, and in any type of analysis like this, we're able to have to do a SNP level analysis using SNP a barcoding approach to check that no samples are duplicated. We didn't want any patients potentially to be enrolled in multiple trials, so we did a SNP barcoding here to ensure that every patient was unique in our meta-analysis. We're also then wanted to benchmark across the platforms. So we were focusing on exome sequencing data, but not all exome sequencing data is alike. We had multiple different capture kits from Agilent and other manufacturers. So we made sure to benchmark things like the number of mutations detected in each cancer type wasn't platform specific. So no bias there in terms of platforms used. And so going back to the results, we're able to quickly identify from the meta-analysis, potentially the most important predictors of this new type of treatment. For example, the number of mutations in each tumor, we found consistently and robustly predicted response here with the strongest effect size, also strongest p-value, as well as key markers, chemokine markers here, CXCL9, which emerged as a very strong effect size predictor of response, better in fact than any other signature. Many RNA-seq signatures have been derived that actually included things like CXCL9, but we found they were really typically overfitted or platform specific, and we're able to identify a much simpler approach of measuring the key chemokine there by itself gave more reliable prediction of response. Really, what was also important in this study was the biomarkers that failed to validate. Probably more failed to validate than did, and of course were limited by our own sample size here. Although this was bigger than any prior study, it's still not as big as we would like. So there's potential some of these biomarkers may have weak effect size that we can't yet statistically detect, but many of them, here had very little evidence of significance. For example, one here, prior reports of the number of copy number changes, somatic copy number alteration burden, that had been previously associated with response to checkpoint inhibitor treatment, published here in very high impact papers here, work in Science, highly cited. Many conferences refer back to this work, and we really saw very little evidence of that as a reproducible predictor, at least one with strong effect size in the study we were able to conduct. We look back at the prior work from those authors, and you can see indeed that when you split patients in a survival analysis into high or low burden of this particular biomarker, you do split the survival curves. Here, patients with a higher burden have reduced response to checkpoint inhibitor treatment across different studies here. And we did investigate this further. We looked in another cohort of samples with checkpoint inhibitor treatment here from the Project Genie initiative. And again, you can split the survival curves. Here, high burden had low response, but you can also have the exact same pattern with any treatment. With non-immunotherapy treated patients, you again split the survival curves, there. So we're able to really conclude that this biomarker, it's either mainly prognostic or it's prognostic and weakly predictive, but it certainly doesn't stand out as a major predictor, you know, that should be considered in terms of clinical utilization for checkpoint inhibitor therapy. We're also looked in this work at the differences in different treatment, different drug or different cancer types scenarios here. We saw some slight differences with biomarkers that were specific only in certain drug situations here. HLA divergence, for example, which measures the diversity of our class 1 HLA supertypes here. You can see that was important, particular therapeutic context here of anti-CTLA-4 treatment. We also knew and were able to quantify the correlation between biomarkers. You know, many of these biomarkers are redundant. They're highly correlated, so the networks here of correlation between measures of mutation burden, different measures of immune cell infiltration, they're all co-correlated with each other, but then, you know, we do see fairly weak correlation between these sort of clusters here, so that there may be some potential utility to combine biomarkers to make a multivariable predictor of response. By having individual patient level data, we could also look for interaction terms here. So we did interaction term analysis. We found several of the biomarkers may be potentially enriched in certain settings. So the mutation burden, for example, had a significant interaction term with histology, you know, much higher effect size in bladder cancer, for example, compared to melanoma here. Similarly, CXCL9 was significantly higher, again, in bladder cancer compared to melanoma. This particular genetic event here, SERPINB3 mutations, looks specific to anti-CTLA-4 treatment rather than anti-PD-L1 treatment. These require further validation, but with individual patient level data, we can at least do this analysis to find preliminary candidate interaction effects. And then lastly, in this work, we were able to start to combine the biomarkers to see, once we'd identified the ones that were important individually, that could be reproducibly associated with checkpoint inhibitors to response. We're able to combine them together in terms of a potential multivariable predictor that may have some clinical utility. So we took all the biomarkers that had validated in the meta-analysis, trained a predictor in the 1,000 patient cohort here using a machine learning approach. The different predictors here that had the strongest importance scores in an XGBoost model here are shown, and then we're able to go on then in 3 different independent test cohorts that hadn't been used in any of the training data to show, at least this is still only retrospective analysis, so it has that limitation, but in a retrospective setting, it had higher AUC values than the current FDA approved biomarker for this type of treatment, tumor mutation burden here. We had significantly higher AUC in a pan-cancer study here from a keynote trial run by Merck. And then in a melanoma, an academic trial here, again, significantly stronger AUC values. And then also another academic non-small cell lung cancer trial here, again, significantly higher AUC performance. And now we can start to take this forward for prospective validation. And so just to say finally here, this is obviously just more of an applied example of using these data sets to get the most out publicly available data. So this is not a formal webinar on actual meta-analysis. You know, obviously that's out of the scope of here. And obviously for those interested in those sort of approaches, things like PRISMA is obviously the place to go to in terms of understanding formal guidelines and additional topics not considered here, such as publication bias, sensitivity analysis, study heterogeneity, etc. So thank you very much for your attention. And then in the last few minutes or so, there's time for any questions.
Thank you, Kevin. Mallory has a question.
Yes, actually, that was the perfect follow-up talk to my talk. Kind of touched upon so many things, and was like the natural next step is actually doing the analysis. And um, I had a question about, so you mentioned, pulling in data from different archives and doing sort of joint or meta-analyses. And I'm wondering how much curation work I don't know how much you were involved in all of those studies you mentioned, but how much curation work was kind of involved in harmonizing data from different places to be able to analyze them jointly?
Yeah, no, an awful lot actually, Mallory. It took several years actually to first of all get the data from the investigators. Not everyone is willing to share data, so it took quite a lot of follow-up. And then particularly on the clinical endpoints, we were fortunate to have a number of clinical oncologist collaborators in this study to really harmonize or find the right response endpoint, more focus on radiological response rather than overall survival, which of course is important, but it potentially more complex endpoint that could be confounded by other factors. We had to sort of harmonize the clinical data extensively and consult with the oncologist to find the most relevant one. The sequencing data is really difficult to deal with. Simple things like the downloading process, you try and continually iteratively download data sets, but it can keep crashing and there can be truncated files. You have to go back to the authors, and some authors had got their sample labels all mixed up, and normals were tumors and tumors were normal. And that was even a major pharma company done that. So the curation took years, and it's probably the most important step in it. But then it means that you can have a sort of legacy from the data and use it for more and more in-depth biological analysis.
Okay, thank you. Dawn, raise your hand as well.
Clapping.
No?
I was second applause.
Okay. Any other question? Thanks, Kevin, for this really a very nice step-by-step guidance, really how to get these data and how to analyze them. Obviously, it is easier for a bioinformatician, I guess. For a non-bioinformatician, is there easy way, or are there easy tools you think that can be used? Because downloading the data can really be daunting, let alone analyzing them.
Yeah, no, I think you probably always would need some technical support in the process, but I think bioinformatics is becoming easier actually, certainly with Nextflow pipelines that are available that are excellent, and also more and more switch to Dockers and container-based deployment of tools. So new bioinformaticians can get started much quicker. Once you've got your basic Linux and you can use a cluster, you can pretty quickly do a lot of the things. Certainly, we'll go back to 5 or 10 years ago, it was much harder. So I think it's getting easier. You still need some training, but people can pick it up. It's no more technically demanding than a lot of experimental protocols, and the documentation. GitHub's becoming much more widely used. So I think it's getting easier.
Okay, thank you. I guess this answers to the question from the chat. What advice would you give to a PhD student hoping to do something similar for the first time? So obviously, if you are not a bioinformatician, the aim of these workshops, really, NIHR statistic group, is to dissipate the myths and to make people who work in a lab or who work statistics to come together and see that if you can learn these protocols in the labs, you can surely also invest a bit of time in statistics or maybe bioinformatics and have a good start in these type of areas.
Yeah, no, exactly. I think for a PhD student, really just come up with either with your supervisor, come up with a question. That's probably where you've got the deep knowledge of what is the most interesting question. And then just start with searching for data sets, define your criteria, and start to work through the sort of process as defined there. If you've got a local statistics support in your institute, otherwise groups like this, I'll post my email in the chat if anyone's working in this area and has any questions. We always love to collaborate with people. So really it's much easier than it ever was before to get started.
Fantastic. Any other question? So coming to your example of pan-cancer biomarker analysis, I really liked it, but it was quite heterogeneous. The data themselves, the cancers, the treatment, some biomarkers might be restricted to certain cancers, but you were only interested in those very common to all, and common also maybe to all, to both treatments, anti-PDL and anti-CTLA-4. So did you consider also stratifying on certain subset which look more alike?
Yeah, no, that's exactly, I think, the limitation or the compromise you had to make with our work is we had to assume a certain amount of homogeneity in cancer type and checkpoint inhibitor. We were limited in some ways because, you know, we didn't have enough data to do meta-analysis in each individual setting. You know, in the future, we'd hope to have a dozen data sets for lung cancer, anti-PD-1 treatment, let's say, but at the moment we've only really had one or two. And so we were stuck with a problem of, you know, the differences across cancer types. Is it to do with biology or is it just study specific factors? And the prior biomarkers in this area, the FDA approved biomarker tumor mutation burden, that is approved across all cancer types as a pan-cancer biomarker. So I think the biology and the prior biomarkers supported our approach that there is value to look at biomarkers that may be important across all cancer types and checkpoint inhibitors. So that's how we ran the first analysis. We did break down where we had enough cohorts, 2 or more cohorts in a particular combination of treatment and or cancer type. So melanoma anti-CTLA-4, melanoma anti-PD-1. We did do sub-meta-analyses in those settings. And to be honest, probably 90% of the biomarkers there were identical to pan-cancer ones. There were some differences. So again, that probably further supported that probably the majority of the biology is the same, but hopefully as data sets expand, there will be some exceptions. And as the data comes, we're very keen to do individual sub ones and not have to rely on a pan-cancer approach.
Okay, thank you. This answers in part also about this other question about heterogeneity between a data set. I was going to add, did you find any relevant clinical metadata to adjust in your analysis, or it was just too messy and too heterogeneous?
Yeah, we did. We gathered all the clinical metadata we could find, and certain things like age, I think we had, and sex we had. Obviously, we could infer from the genetic data. Obviously, also ethnicity we could infer from the data. Stage wasn't universally available, but these were all metastatic stage patients. There hadn't been any data from neoadjuvant earlier stage immunotherapy trials at that point. So they were all late. We didn't have stage, but they were all late stage. So that wasn't probably a major issue. We would have liked to have had a lot more things, ECOG performance or tumor volume, but it just wasn't there consistently enough.
Yeah, thank you. So there's also a question whether it is okay to share the slides. Just for the record, the webinar is being recorded, so it will be accessible after today.
Sure.
Slides maybe can be shared. Mallory has shared hers.
Yeah, definitely, yeah. Okay,
thank you. So we are having a short coffee break now. Can we reconvene just before 11 for the last talk? Thank you. Okay, thank you for staying till the last talk. After introducing others, let me introduce myself too. So I am Jeremy Sengimana. I work at Newcastle University. I was trained in Belgium, University Catholic of Louvain, the other UCL. So there is UCL here in the UK, but there is one there in Belgium as well. So UCL for University Catholic of Louvain. I have worked many years at the University of Leeds, where I was lucky enough to be involved in generating, analyzing, and archiving large-scale genomic data, and also accessing data from public repositories. So I'm really glad to share with you some lessons learned on clinical metadata accompanying these large-scale genomic data. So the outline is as advertised. So I will go through what I mean by clinical metadata. Why do I think it is a rare commodity? You may disagree, so we will have a time to share views about it. So why high quality metadata is even rarer if we are looking at public repositories, and then some concrete examples of where things can go wrong. So what is clinical metadata? For the purpose of this talk, I mean by clinical metadata, a set of variables or phenotypes or of clinical relevance, generally collected on study participants alongside the molecular data. These include primary and secondary outcomes of interest, as well as predictors, confounders, all the variables we commonly refer to as covariates in analysis. Typical examples include some demographic information, such as those already mentioned in the previous talks. We have heard about how it is really tricky to code a simple thing as sex. It includes some histopathology data, some environmental exposures, some medication, and so on. So it is variable per study. The level of completeness is obviously very variable, and it is a rare commodity for many reasons. Data collection is tedious. Some of these variables come from self-reporting. Others are extracted from clinical notes. As we know, this is are not homogeneous. Some come from registries, and some have constant updating from follow-ups. There are no common standards really, or platform like it will be for molecular data, or minimum number of variables required when you submit your data. And there should be some harmonization, but this is really tricky to do. It is not as easy to cost any projects. Again, if you compare it to molecular data, well, there are a few platforms, and you contact the service providers maybe, and you get some quotation before you submit your grant application. Factoring in the metadata is more difficult. Sharing regulation or guideline are looser than for molecular data. Owners might not see any incentive to share them, particularly if they are still using them themselves in their projects. And if they feel that the data needs updating, and if you submit it, then the update will be lost to the downloaders. Identifiability can be an issue. Some combination of particular variables can make an individual identifiable, particularly in the rare categories. So in the end, sometimes the best solution is really to contact the data owner. And this is where the controlled access Mallory has mentioned can be very helpful. So if you download the data from public space, having high quality metadata is really tricky. An example is when you have, for example, submitted metadata after analyzing them and having one publication. So the kind of quality control you applied might be good enough for that particular manuscript, but not for further studies. So more QC might be needed, but all people who download these data might not be comfortable or even feel that they have enough time to do it. So that's why there are quite of issues in many publications. I will highlight shortly. So failing to acknowledge that could affect your analysis, obviously, in the interpretation, and there can be misuse of data dictionaries, which can cause misleading conclusions. So let's illustrate this concept with an example of a very popular data set, the TCGA. TCGA stands for the Cancer Genome Atlas. It has started in 2006 as an initiative of the NCBI and the National Human Genome Research Institute in the US. At the moment, it has a lot of molecular data and metadata, obviously, for matched normal tissue and 33 cancers, for more than 11,000 patients, 7 types of molecular data like gene expression, like mutations, copy number variation, etc. About 20 institutions in the US are involved. The data generated is cover more than 200,000 DVDs, and it has been extensively used, and it has had a great impact really on biological and bioinformatics research. These are listed on this website to show how this cohort, this resource has changed really the world of cancer research. A quick search on Medline on the citation of TCGA shows that since the first few publications citing this resource, then there has been an exponential increase over the years. These probably only include those citing TCGA in the title or the abstract, not the whole body of the publication. However, my point today is that some of these publications may have misused the metadata. And that is because tracking the origin of each sample profiled for molecular data is not really obvious. So this shows an example really a participant might have diagnosed in one center and their tissue and metadata sent to another center for profiling, for example, gene expression profiling. And you can see from this cartoon that the sample might have come from a tissue from one particular site, but it was partitioned in many sub-samples, the so-called vial, the portion, the analyte, and in the end, put on a plate, different place of any plate here, and send it to a different center. So being able to track each particular molecular data and match it with the correct metadata is not as easy as it seems, or at least all the data users don't necessarily take the careful approach to that. This is the barcoding of each particular sample in TCGA, showing the different information. So this is the site where the participant was recruited. This is the center where the sample was profiled. So the plate, the portion, the subsample, etc. So there could be difficulties in matching the molecular in the metadata for many reasons. Certain types of metadata relate to the patient, while others relate to a small portion of the biopsy analyzed. So molecular profiling might have used a biopsy taken at the time of relapse, for example, but available metadata related to initial diagnosis. And data users don't necessarily always check this discrepancy. The cohort has been followed up for a very long time, which means there has been multiple changes in technologies and tools. So there is a lack of homogeneity, and there could have been some retrospective updating, but this is very limited, as I will show on one example. So all in all, caution is needed where linking certain measurements. To make things less abstract and more concrete, I'm going to talk about the example of skin cutaneous melanoma. I will show the different disease staging version used. So there is one tool called AGCC staging that has been evolving over the time. So different editions were used in different patients. There is also what look like a misleading tumor stage information when you compare the tissue that was profiled for molecular data and the corresponding AGCC stage. And there is also a well-known really issue with heterogeneous recording of survival data, which has a big impact on how to interpret survival analysis. So a quick word on the AGCC staging. AGCC stands for American Joint Committee on Cancer. It is a committee that proposed a way to stage cancer. The current edition is number 8 from 2018. So edition 1 started from the 80s. It is based on 3 components, the T, N, and M components, which I'm not going to go into details. What is important really to remember is that across all the editions of this staging tool, stage 1 and 2 melanoma are localized. They are primary and localized in one place. Stage 0 is when it is really carcinoma in situ. Too small or to be staged. And when stage 3 and 4 are used, this is when the disease is metastatic. Stage 3 is when the disease has spread to at least one region or node. And the stage 4 is when the disease has reached a more distant organ like the lungs or the brain or the intestine, for example. So 3 and 4 are for metastasis. Now, the first problem in the CGA metadata is they used different AGCC staging editions, as I just mentioned. If you plot this pie chart on the tissue that was profiled for genomic data, you can see that just under half of them, there are for melanoma about 470. Just under half of them used edition 7. Remember, the current edition is 8, so it has not been used. But worryingly is that edition 6, 5, 4, all the way to 1 have been used as well. This is not surprising because if you plot the edition used and the year of diagnosis on a vertical axis here, you can see that in the 80s, those diagnosed in the 80s, they used the tool that existed then. So that was edition 1. And the more recent ones obviously have used the more recent staging edition. And there has been also an attempt to, as you can see here, to upgrade those old recruits, old diagnoses using the newer version. But we have still a lot of heterogeneity. This means if a cancer is staged as 2, for example, or 1, or the subgroup of these, because there is also A, B, C, D, 1A, 1B, 1C, 2A, 2B, 2C. All these will not be consistent across different edition. And then if you adjust this covariate in your analysis, like many publications do, it will not be correct. Problem number 2 is that stage shown in the data may even be misleading, may be unrelated to the tissue used for molecular profiling. So you have to make a bit of investigation for that before you adjust that covariate in the first place. How do we know that? Well, when you look at the metastatic subsets of melanoma, they should be all staged 3 or 4, but you see all the stages used. So 3 or 4 are shown here on the left. They represent less than half. So 1 and 2 were used when we know it doesn't correspond to metastasis. So there is an explanation, I think. That's because the biopsy staged is not the one profiled. There is nothing wrong in the way data are recorded, but it is just misleading for the users, as is often happen. Stage was given to the biopsy of the initial diagnosis, which may or may not be the one profiled. It has not been subsequently updated. Problem number 3, we see that when you look at a plot like this, the primary tumors profiled for molecular data seem to have been come from patients diagnosed much more recently. So around the year 2010, while the metastases were diagnosed all the way from the 80s to 2010, 15. So this is a bias which needs to be borne in mind. If you consider to the right-hand side here, for example, where there is no bias, this is how it looks like. So there are males and females diagnosed all the way across the lifetime of the period we are looking at, which is not the case for the tumor subtype. And as a consequence, and this is really a big issue that we found in the data and is very rarely acknowledged or reported. If we use this survival data for survival analysis, what you see is that quite surprisingly, patients with metastases would appear to have a better prognosis. So this for those who are not familiar with this plot, this is a Kaplan-Meier curve showing the difference in the survival between these two groups. So on the horizontal axis, we have the follow-up time from time of diagnosis, which is 0. And on the vertical axis, we have the proportion of patients who are still alive. So at the beginning, at diagnosis, 100% are alive. And then as time elapses, the number of those still alive decreases. So you can see that after 5 years, those still surviving among those with primary cancer is only 20%, while it is in the 70s, 70% in metastases. So this is plainly wrong. And when people use this data to find biomarkers and use primaries and metastases together, they don't take account of this aberration. So the difference is very highly significant. You can see the confidence interval don't overlap. So why do we see this? It is because primary and metastasis in this context refer to the biopsy used to generate molecular data. And that may differ from the initial diagnosis. So most metastases profiled for molecular data appear to have occurred as a recurrence many years after remission. This is shown by the AGCC staging. Many of these have stage 1 and 2, as we just said, which means they were not metastases at the beginning. But this information is rarely used in any analysis of this cohort. The follow-up time was recorded since the very first diagnosis, not the recurrence. Now, for primaries, for those tissue recorded as primaries, the follow-up time look correct. There is no bias from recurrence. There is no bad follow-up start time. But it is worrying to see that there are so many early deaths. So we have a survival rate after 5 years of only about 20%, when we expect 95% in primaries in general. So why do we see this? It is because these are not representative of the population. They are very highly selected for the most advanced tumors. The very thick ones, at 49 out of 104 have more than 4 millimeters of thickness. This is in the very thick category. And even 31 out of 104 have stage 3 or 4. That means they had metastasis at diagnosis. So although the primary disease, primary biopsy was used to generate molecular data, the patient had also a metastasis. So it would be wrong to suggest that the survival profile we see here is reflective of the nature of a primary tumor. So to conclude then, this is my last slide. It seems like CGA melanoma primary and metastasis, maybe other cancers as well, I have not looked at other cancers, should serve to study different questions. Really, there are two different cohorts, and pulling them requires extensive exploration and justifications, which are very lacking in many publications. Using highly selected samples is probably okay for cancer biology studies to understand molecular mechanisms, etc. But it seems to me that it is problematic when you use them for epidemiological studies where you would hope to represent the population, and for translational studies where you can find interesting biomarkers, but you don't know exactly how many people would benefit from that biomarker. And besides, adjusting metadata is tricky in this type of data because it is so heterogeneous and difficult to harmonize. Metadata misuse can be caused by simply not giving it enough time to explore, to understand what the data mean, or lack of statistical and epidemiological skills. And surprisingly, this is not only due to the users not being able to. It can also be caused by the reviewers who suggest to use a given data set, which is very well publicized, which is well known, and I have seen many authors trying to use this resource, TCGA, just to accommodate a request from a reviewer. In general, the way the data are recorded corresponds to the way it was collected, and maybe the way it was used in the first place, the first publication. So doesn't a priori has anything wrong, measure which is wrong, but it is just important to understand it before using it, to understand its limitations, to know what can be done and what cannot be done. Obviously, some harmonizations would be good, but they cannot, to my mind, be done at the end users by the downloaders of the data. It has to be done by the submitters. So this is what I want to share with you today. Thank you for listening, and I think you have time for questions. So, Mallory?
Thanks for that great talk. It was a really interesting use case for a data set that has potentially had lots of value, but also has maybe these things that we should be considering in more detail. I'm wondering if you know what kind of community effort there has been to take feedback like the example you showed, or other examples of where misinterpretation could be easily done, and feed that back into whoever's maintaining the data and say, like, look, perhaps we need to do another round of curation or make other information available to help prevent people from falling into some of those pitfalls you mentioned.
Okay, thank you. Very interesting question. So the first time I noticed the seemingly protective effect of metastatic cancer, it was quite shocking. And I was trying to do a given analysis, obviously, and I stepped away from doing it. I tried to share this with many other people. Actually, I thought I did something wrong by my own analysis. I re-downloaded the data to see the most updated version, and then I also asked other people to check if my analysis was correct, or what they get from their own downloaded data. Obviously, we download them from the same repository. So yes, it can be misleading. I haven't gone obviously through all the publications to see who has used it as it is, who hasn't. But I have seen a few, not because I was after them, but just because I was interested in them and reading them and noticed. Or when I review papers, I very regularly see this. And I have had to push back on some reviewers asking me to use this data in the way they were suggesting, sharing with them my experience with it, and yeah, many have come to realize it. But yeah, this data is very heterogeneous, and it was submitted as it is, and you know. There are attempts to harmonize molecular data, for example, with different builds of human genome. Some data have been, upgraded, etc. But for metadata, I don't know. They just assume the data users will take all the due care to use them in the way it is intended to. But a lot of people will just download it and adjust age, sex, stage, tumor type without trying to explore more. Dawn, is your hand raised this time?
Yes, I've really got my hand up this time. Thank you. Yeah, I mean, that was really nice. The three talks together today have highlighted how important it is. This is really expensive data that's being collected here, and it makes a lot of sense for it to be shared, but all the difficulties that come with that. And if you just naively go in to a data set, you're really, really likely, I mean, it's true in any context, isn't it? Any data set, you might just naively go into it, you're going to probably misunderstand something. But here, due to the sort of the nature of the data, it looks, because of the way that you've collected the different components of data for different purposes, it's a bit more fraught. And I think it contrasts quite nicely with the research parasites reference that Kevin brought up, which I wasn't aware of, but it just shows actually no, it's not, it's not that easy to just come in and analyze other people's data. It does have to be done really properly. So it's sort of made me realize that yes, this is quite complicated stuff to grapple with. So it's really nice to see. Well, I wondered actually, have there been any sort of famous examples where there had to be any retractions because of some, naive approaches?
Not to my knowledge. Maybe others might be aware. Heather?
Just before I stop, actually, Jeremy, there are some questions in the chat that after Heather's question, we could go to the chat. Thanks.
Thanks, Jeremy, for a nice talk. Just coming back on Dawn's point, the only couple of retractions that I'm aware of, which are kind of old examples, well, one of them certainly was in the early days of GWAS. There was the longevity GWAS, where I guess the people analysing it were more statisticians and not so familiar with the kind of QC that you have to do for SNP arrays. So that ended up getting, well, the original paper was retracted. I guess they then did some subsequent analysis with a bit more care or a bit more advice from people who knew the field. So that was one example. Anyway, the question I had was, so your example, Jeremy, was very much, I guess, I mean, it was supposed to be a general point, but you were giving the example of cancer, where you had this issue to do with the primary and non-primary and so on. And that just highlighted to me that, you know, a lot of these issues are going to be quite field specific. I mean, that's a very cancer specific issue. There are going to be similar issues if you're working in different clinical data sets, different diseases, different types of question. And does that mean that it's really quite important to either become an expert in that area or collaborate quite closely with experts in that area to be able to even understand the metadata and understand what the issues might be that you'll have to think about?
Yes, absolutely. So the key message, I guess, is like we always say in statistic, it is not about trying to find the significant p-value straight away and then report it. It is important to really understand what you are doing. If it is tricky and it is a different field, obviously, and we collaborate with people from different areas. It is an investment in time for you to understand it, but it might be easier to collaborate with someone who knows, who can help out in understanding quickly what these things mean. So someone asked about breast cancer. Is it the same TCGA breast cancer? I don't know. I haven't checked, but what you really need to know is it can be easy to get the data and easy also to extract the kind of information you want. But it can be just too comfortable to do that. And I just noticed it without investigating it for this particular example. I'm sure you will see in many papers adjusting covariates in using TCGA data and yeah. So the most really aberrant, I think, is even when you know it is a primary that has been profiled, there is an information that is hidden in the background that the patient had also a metastatic at that time, but there is no genomic data for it. So one of the questions, are you saying that primaries have been misclassified because of samples were taken before metastasis occurred? Yes. So my understanding really is that the survival information, the follow-up of patients is from the very first diagnosis. So those who were diagnosed in the 80s, you have to be aware that the TCGA started only in 2006. So they were included in the study retrospectively. And seeing that they survived 25, 30 years with a metastatic cancer is very surprising. And then when you dig a bit, you see that the cancer they were first diagnosed with was not a metastasis. Otherwise, it will not be stage 1 or 2. And then you see that if it was stage 1, it was probably removed, and then relapse occurred like 10, 15, 20 years later. Any other question? So if there are no more questions, then I suggest that we go in breakout. Alice or Dawn, can you? I guess we can have say three breakout rooms, and each one join one they want. And
Jeremy, I've handed over the host to you. Actually, I can't stop the recording, so I don't know if Dawn or Jeremy, you'd like to stop the recording at this